Los pongan en modo silencioso, por favor. Antes de dar inicio, les suplicamos su atención para observar el video de Protección Civil con unas breves instrucciones de seguridad. Gracias por ello.
Este video le mostrará las medidas de seguridad y acciones a ejecutar si ocurre un sismo y usted se encuentra en el interior de uno de los 3 auditorios de la Facultad de Medicina de la UNAM. El auditorio Raúl Fournier Villada tiene una capacidad para 971 personas y cuenta con 10 salidas de emergencia. 6 se localizan en la planta alta, 4 a los costados y 2 en el pasillo central, mientras que la planta baja cuenta con 4, 2 en la parte posterior y 2 en los costados, más las 2 entradas principales por el centro. Es importante recordarle que en caso de siniestro se debe guardar la calma, no correr, no gritar y no empujar. En caso de sismo, usted debe esperar a que éste finalice y recibir indicaciones de los brigadistas de protección civil, identificados con este chaleco naranja, quienes los guiarán a las zonas de seguridad fuera de los auditorios. Por último, le pedimos que siempre traiga consigo una identificación personal. La Facultad de Medicina de la Universidad Nacional Autónoma de México le da la más cordial bienvenida a esta, su universidad.
Sí, señores, muy buenas tardes tengan todas y todos ustedes. La Facultad de Medicina de la Universidad Nacional Autónoma de México les da la más cordial bienvenida a la conferencia magistral que dictará el doctor Drew Weissman, Premio Nobel de Medicina 2023. Nos acompañan en el presidium, por supuesto, el doctor Drew Weissman, Premio Nobel de Medicina 2023. La doctora Ana Carolina Sepúlveda Vildósola, directora de la Facultad de Medicina de nuestra universidad. Y el doctor Raúl Carrillo Esper, presidente de la Academia Nacional de Medicina de México. Agradecemos a todos los asistentes, autoridades universitarias, presidentes de academias y asociaciones, titulares de institutos nacionales de salud, directivos, académicos, investigadores, alumnos de nuestra facultad y demás invitados que hoy nos acompañan en este auditorio, así como a todas las personas que nos siguen a través de las redes sociales de nuestra facultad. Sean todos y todas bienvenidos. Tiene la palabra la doctora Ana Carolina Sepúlveda Vildósola, directora de la Facultad de Medicina de nuestra universidad.
Muy buenas tardes, tengan todas y todos ustedes. Distinguido doctor Drew Weissman, doctora María Soledad Funes Argüello, coordinadora de la investigación científica en la Universidad Nacional Autónoma de México, autoridades de la Academia Nacional de Medicina, integrantes de la Facultad de Medicina, de la comunidad universitaria, estudiantes, colegas, invitados especiales. Doctor Weissman, con enorme orgullo y profundo respeto, le damos la más cordial bienvenida a la Facultad de Medicina de la Universidad Nacional Autónoma de México. Nos honra profundamente recibirle en esta casa de estudios en el marco de su visita a nuestro país y de su investidura como académico honorario internacional de la Academia Nacional de Medicina. Que su presencia en esta facultad forme parte de un recorrido por México, sin duda, para nosotros es motivo de gran celebración en nuestra comunidad. Su trayectoria y su legado científico marcan una de las más relevantes contribuciones a la medicina contemporánea. El Premio Nobel de Fisiología Medicina, que le fue conferido en 2023, junto con la doctora Kathleen Caricó, no solo reconoce una extraordinaria innovación científica, la tecnología del RNA mensajero que dio origen a las vacunas contra la COVID-19, sino también representa una esperanza tangible para la humanidad en su lucha contra múltiples enfermedades. Sabemos que este avance no surgió de la noche a la mañana. El desarrollo de la vacuna basada en RNA mensajero es el fruto de muchos años de trabajo persistente, de investigación rigurosa y de convicción profunda en el poder transformador de la ciencia y ocasionalmente en la serendipia. Su historia nos recuerda que detrás de cada gran hallazgo hay años de esfuerzo silencioso, de colaboración y de convicciones firmes. Permítanme compartir con ustedes una anécdota que el doctor Weissman nos platicó ayer en la noche y que me dio permiso de decir el día de hoy, y que con humor muestra el carácter profundamente humano que también acompaña el trabajo científico. Su hija mayor, una estudiante excepcional, le solicitó ayuda para realizar una tarea de biología en la preparatoria, a lo que él accedió con gusto. Sorprendentemente, el resultado fue que ella obtuvo una calificación de B-, la única de ese tipo en toda su trayectoria escolar. Sin duda, es una anécdota entrañable que nos recuerda que incluso los más grandes científicos pueden enfrentarse a los misterios de la paternidad. Hoy, en este encuentro con estudiantes, profesores, profesoras, investigadoras, investigadores, celebramos no solo su descubrimiento, sino su ejemplo, porque escucharle es una oportunidad invaluable de inspiración para quienes formamos parte de la ciencia médica en formación o en ejercicio. Doctor Weissman, permítame contarle un poco sobre nosotros. La Universidad Nacional Autónoma de México es la institución de educación superior más importante del país y una de las más antiguas de América. Sus orígenes se remontan al 21 de septiembre de 1551, cuando la corona española concedió por cédula real la fundación de la Real y Pontificia Universidad de México. A lo largo de los siglos, nuestra universidad ha sido un pilar de la vida intelectual, científica y cultural de México y una formadora de pensamiento crítico y compromiso social. Desde su autonomía en julio de 1929, la UNAM ha consolidado su vocación como universidad pública, laica y nacional, guiada por los principios de libertad de cátedra, investigación y pensamiento. Es líder en Latinoamérica e Iberoamérica. Nuestra Facultad de Medicina de la UNAM es la más antigua del continente americano. Sus orígenes se remontan a 1579, con la fundación de la Cátedra Prima de Medicina en la Real y Pontificia Universidad de México. A lo largo de casi 450 años, hemos formado múltiples generaciones de médicas y médicos comprometidos con la salud, la ciencia y la sociedad. Actualmente somos la institución formadora de médicos más grande de América Latina y una de las más importantes del mundo hispanoparlante. Nuestra facultad ha sido consistentemente reconocida en los rankings internacionales como la líder en formación médica en México. Este liderazgo ha sido posible gracias al compromiso permanente con la excelencia académica, la investigación científica y el servicio social. Pero más allá de las cifras y reconocimientos, lo que da sentido a nuestra misión es el diálogo con figuras como usted, doctor Weissman, cuya vida y obra encarnan los valores más altos de la medicina y la ciencia. Su presencia hoy fortalece nuestra convicción de que el conocimiento científico debe estar al servicio del bienestar humano y que la colaboración internacional es indispensable para enfrentar los desafíos globales en salud. Nos honra especialmente compartir con nuestra comunidad que el doctor Weissman ha accedido generosamente a establecer una colaboración con nuestra facultad. Gracias a su apertura y entusiasmo, nuestras profesoras y profesores, así como nuestras y nuestros estudiantes, podrán realizar estancias cortas en su laboratorio, donde tendrán la oportunidad de aprender directamente de una de las mentes más brillantes e innovadoras de la medicina contemporánea. Le agradecemos profundamente este gesto de generosidad y de confianza que sin duda marcará la vida académica y científica de quienes participen y de la ciencia en México. Agradezco sinceramente a la Academia Nacional de Medicina de México por haber incluido esta visita a nuestra facultad dentro de las actividades de su investidura como académico honorario. Esta decisión honra a nuestra comunidad y fortalece los lazos entre nuestras instituciones. Doctor Weissman, le recibimos con admiración, con entusiasmo y con un profundo deseo de aprendizaje. Sea usted bienvenido a la Facultad de Medicina de la UNAM. Esta es su casa. A continuación daré una breve lectura a su inmenso currículum. Hicimos una condensación que les presentaré a continuación. El doctor Drew Weissman realizó sus estudios universitarios en bioquímica y enzimología en la Universidad de Brandeis y su grado de doctorado en la Universidad de Boston en inmunología y microbiología. Su residencia fue en medicina en el Beth Israel Hospital de Boston y su fellowship en alergia e inmunología en el NIH. Está certificado en medicina interna y en alergia e inmunología. Su carrera académica la ha desarrollado en la Escuela de Medicina de la Universidad de Pensilvania, donde es profesor distinguido en vacunas y donde imparte clases y ha formado a destacados investigadores. Además, es profesor invitado de la Universidad de Chulalongkorn en Tailandia. Posee 300 publicaciones en revistas de alto impacto y es inventor de una docena de patentes otorgadas en países de América, Europa y Asia. Ha disertado en numerosos eventos sobre la tecnología del RNA mensajero con nucleósidos modificados, de la que es su coinventor y por la que ha sido reconocido con el Premio Nobel, como ya se mencionó, en 2023, junto con su colaboradora Kathleen Caricó. Esta tecnología ha permitido desarrollar nuevos agentes terapéuticos a base de nanopartículas lipídicas portadoras de RNA mensajero contra enfermedades infecciosas como el SIDA, la COVID-19 y la influenza, y recientemente nos compartió otras enfermedades como la enfermedad vascular cerebral, las enfermedades autoinmunes y la leucemia. Actualmente es el director de investigación en vacunas de la División de Enfermedades Infecciosas del Centro Médico de Administración de Veteranos en Filadelfia y del Instituto para la Innovación de RNA de la Universidad de Pensilvania. Su trayectoria de investigación ha estado impulsada por honrosas becas y por cuantiosos reconocimientos en investigación en su país y en el extranjero, incluidos doctorados honoríficos por las más prestigiadas universidades norteamericanas. De entre sus numerosos reconocimientos, resalto el reconocimiento como rescatador de la humanidad y el de Salón Norteamericano de la Fama de Inventores. Es miembro de las Academias Americanas de Arte y Ciencia, de Medicina y de Inventores, y desde el día de ayer de la Academia Nacional de Medicina de México. Les dejo ahora con el doctor Raúl Carrillo Esper.
Muy buenos días a todas y todos ustedes, paz y bien. Y bueno, para mí, como siempre, es un orgullo y un honor estar en la Facultad de Medicina, en el campus de la Universidad Nacional Autónoma de México, como bien lo comentó la doctora directora Carolina Sepúlveda, una de las mejores universidades a nivel de Latinoamérica y yo diría a nivel mundial. Y es para mí también muy grato venir en representación de la Academia Nacional de Medicina de México. Doctora Carolina Sepúlveda, muchas gracias por abrir las puertas de esta grandiosa Facultad de Medicina y de la universidad también. Acabamos de estar con el señor rector a la Academia Nacional de Medicina de México, distinguida comunidad universitaria en la que se amalgaman y acrisolan en este momento toda la comunidad. Docentes, investigadores e investigadoras, todos los directivos y autoridades de la Facultad de Medicina, y bueno, sobre todo que hay una buena cantidad de estudiantes de medicina, lo cual halaga mucho este día. Porque hoy, como ya bien lo comentó la doctora Sepúlveda, es un día muy especial, porque el doctor Weissman, Premio Nobel de Medicina, está aquí en la Facultad de Medicina. Y qué mejor que esté aquí, porque es una de las mejores facultades de medicina de este país y del universo. En ese sentido lo creo yo, por el impulso que tiene, por la doctora Sepúlveda, que le ha dado un hitazo importante y un impulso a esta facultad y a todo su grupo de trabajo, y sobre todo a sus profesores, investigadores, y en especial a las estudiantes y a los estudiantes. Por eso la Academia Nacional de Medicina se congratula de estar el día de hoy. Y qué mejor, porque el doctor Weissman, como ya lo comentó la doctora Sepúlveda, es ejemplo de tenacidad y de resiliencia. en su vida como investigador seguramente no siempre se la vio fácil y salió adelante. Porque ser investigador y un investigador dedicado para desarrollar dentro de sus procesos de investigación algo que vaya a ser en beneficio de la humanidad, hay pocos. Porque muchas veces la soberbia puede desviar el camino de un investigador o de un médico clínico. Pero en el doctor Weissman se ve cómo este esfuerzo tuvo un objetivo social. Y gracias a su desarrollo, doctor Weissman, millones de vidas humanas se salvaron en este planeta. Y con todo el desarrollo que está haciendo en su laboratorio, con todo el grupo de sus colaboradores, va a llegar este desarrollo del RNA mensajero para tratamiento de diferentes enfermedades, como ahorita bien lo comentó con el señor rector, va a ser espectacular. Lo que viene al mundo en los siguientes años se debe a gracias a personajes como el doctor Weissman, que es vanguardista, visionario, su grupo de trabajo, la integración de equipo, porque todo esto no es de una sola persona, es de un gran equipo de trabajo. Y esto es muy importante para que las chicas y chicos jóvenes que están aquí, estudiantes de medicina, aprendan esto. La medicina tiene que ser transdisciplinaria y multidisciplinaria. Tienen que aprender a trabajar en equipo, que la soberbia no les nuble la vista. Es un trabajo común y de muchos. Por eso es muy importante el día de hoy aquí para la Academia Nacional de Medicina. Y para hacer esto, tiene que siempre haber alguien detrás que haga la magia, porque la venida del doctor Weissman, además de su bonomía y generosidad, fue gracias al trabajo incansable, puntual y obsesivo del doctor Hugo Barrera Saldaña. Gracias, Hugo. El doctor Hugo Barrera es un investigador de altos vuelos en nuestro país. Como yo digo, es un SNI Super Plus Ultra. Pero además de eso, al igual que al doctor Weissman, lo caracteriza su sencillez, bonomía y gentileza. Gracias, Hugo. Y este reconocimiento que hago aquí público es aquí, con la comunidad universitaria, porque Hugo se ha desarrollado también dentro de la comunidad universitaria. Él es originario de Nuevo León de Monterrey y trabaja en el Hospital Universitario de Nuevo León, un gran hospital. Y bueno, felicidades, Hugo, y muchas gracias. Este reconocimiento de todo corazón. Un aplauso para Hugo. Y bueno, y también agradecer a la doctora Carolina Sepúlveda, al doctor Enrique Graue, que somos parte de la mesa directiva de la academia, porque esto se debió al trabajo de todos. Cada quien puso de su parte lo que tenía que hacer y se logró. Entonces el doctor Weissman está aquí con nosotros. Y esto es un impulso a la investigación mexicana, porque la investigación mexicana es parte fundamental, es el motor del desarrollo del país. Un país sin investigación, un país que no desarrolle patentes, un país que no esté a la vanguardia, un país que no compita a nivel internacional, es un país muerto, es un país que se acaba, es un país que no va a dejar ningún legado a las siguientes generaciones. Y la imagen del doctor Weissman es para seguir impulsando a través de un trabajo colaborativo que está muy abierto el doctor Weissman a hacerlo. Gracias, doctor Weissman. Bueno, ya la doctora Carolina les comentaba, está muy bien predispuesto a hacer esos canales de unión. Entonces va a haber rotaciones y muchas cosas más que van a aprender ustedes. Entonces esto habla también de la bonomía del doctor Weissman. Doctora Sepúlveda, muchas gracias por la apertura aquí a la Facultad de Medicina y a la universidad y estar aquí en este auditorio. Por este auditorio y por este podium han desfilado desde su fundación las más grandes y los más grandes pensadores, no solamente de la medicina, los investigadores, sino también de las ciencias, de las artes, del humanismo y del humanitarismo, que están muy bien entrelazadas con el desarrollo médico y el desarrollo de esta facultad. Y el nombre que lleva este auditorio es increíble, el doctor Raúl Fournier Villada, gran académico, miembro de la Academia Nacional de Medicina y en su momento presidente de la corporación. Y esto también es gustoso para la academia, que lleve el nombre de este auditorio, de uno de los grandes de la medicina mexicana, vanguardista y con una gran visión a futuro. Así que por todo esto estoy muy contento. La academia, les repito, se congratula de esta invitación y también agradecer al doctor Weissman, ya investido como académico honorario. Él es el 20º Premio Nobel que ingresa como honorario a la corporación. El primer Premio Nobel que ingresó fue el doctor Roberto Koch Y de ahí le siguieron otra serie de grandes investigadores que han fortalecido y han impulsado la medicina, la ciencia, la investigación para el bien común. Así que, bueno, doctora, muchas gracias. Doctor Weissman, muchas gracias. Gracias a todas y a todos ustedes que se tomaron unos minutos de su muy apretado tiempo y agenda para estar aquí en esta ceremonia tan importante. Que tengan muy buen día y por mi raza hablará el espíritu.
Señoras y señores, tiene la palabra el Premio Nobel de Medicina 2023, el doctor Drew Weissman.
Thank you very much. I'm highly honored to be here and to be made a member of this university, an invited guest, and hopefully a future collaborator. So what I wanted to talk to you about today, I'll start off giving a little bit of background on how mRNA was developed. And then I want to talk about what I see as the future of mRNA, which is also a future of medical therapeutics. I always start off by showing this timeline to give people an idea that even though everybody said RNA was invented in 10 months, it really wasn't. RNA has been around as long as I have. It was developed in 1961. The first time it was injected into people as a clinical trial was in the late 1990s. So RNA has been in people for decades. The first time it was injected into an animal with the idea of using it as a therapeutic delivery system was in 1990. That was followed by studies using mRNA as a vaccine. Now, it was an unusual vaccine. What they did is they would take dendritic cells out of people, pulse them with mRNA of different designs and manufacturers, and then inject the dendritic cells back in. That's because when they injected RNA in, people got sick. The reason for that, we now know there are 17 innate immune sensors. These are germline encoded sensors that recognize different forms of RNA. And when they bind RNA, they produce inflammatory cytokines, type 1 interferons. They can directly inhibit ribosomal translation. They induce apoptosis and cell death. So when Katie and I started working together in 1997, the pharmaceutical industry, the biotech industry, the NIH, pretty much everybody had given up on RNA. It had failed clinical trials that showed no efficacy in the cancer vaccines being used for. And everybody said, oh, it's too inflammatory, it doesn't make enough protein, it's too hard to work with, don't bother with RNA. Katie and I did anyway. One of our telling experiments that led us to our hypothesis was when we took RNA, either from bacteria or mammalian cells, and isolated RNA from different organelles or different regions of the cell. And then we added that RNA to dendritic cells. Dendritic cells are the chief antigen-presenting cell. Their job is to go around and collect foreign things and bring them back to start immune responses. Dendritic cells have most of those 17 innate immune sensors. And when we did that, we had some very interesting results. What we found is that transfer RNAs had the lowest amount of innate activation, and bacterial RNAs were nearly as high as in vitro transcribed. So that led us to a hypothesis. Our thinking was that transfer RNAs can have up to 25% of their nucleosides modified. So nucleoside modification occurs in every organism, from bacteria through mammals and everything in between. There's over 100 different types of nucleoside modification. We don't know what many of their functions are, but some are conserved. So what we had to do to test our hypothesis is we had to make nucleoside modified mRNA. Most of the enzymes to do this weren't described yet and wouldn't work because they work in an RNA-dependent manner, which means they modify at a specific site. So what we did is we tricked the phage polymerase that makes RNA from DNA into incorporating modified nucleosides. So what you see here is every uridine is now a pseudouridine. Every cytidine has a 5-methyl in this mRNA. And what was striking was that any uridine modification got rid of most of the inflammatory activity of the RNA, but not modification of other nucleosides. Now, the next experiment was even more striking. Katie and I had a bet, and the bet was whether RNA would be translated when it's fully modified. And we both said no, not a very good bet. Not only were we wrong, but we were wrong tenfold in the other direction. The modified RNA was translated much better than unmodified RNA. We also found that the phage polymerase that makes RNA makes errors of double-stranded RNA. And if we got rid of those contaminants, we could increase RNA translation by 4 orders of magnitude. So in our minds, that addressed 2 of the chief difficulties that pharma and biotech and NIH had identified. RNA is too inflammatory and it doesn't make enough protein. So we're now looking at the early 2000s, and I'm an HIV researcher. I did my fellowship with Tony Fauci doing HIV research, and I would go to HIV conferences around the country, and I would present our RNA work. Typically, I was the only person working on RNA. And afterwards, I would sit down and talk with Tony and talk with Gary Nabel and talk with other experts in the field. And they would ask me what I'm doing, and I would tell them I'm working on mRNA. These are our thoughts. And they would smile and they would nod. And they would sometimes say, you know, you really need to switch to something else. RNA is never going to be useful. It's never going to work. You're wasting your time and you're wasting your career. So I would try and argue with them, and I'd say, you know, protein therapeutics is the most rapidly growing element of pharmaceutical development. This is what it takes to make a monoclonal antibody. You start with a 50,000-liter drum of CHO cells or other cells. You then have to invent a purification procedure to purify that monoclonal away from the serum proteins. So what that means is that every new monoclonal antibody, you start from scratch. And in the United States, the cost is between $50,000 and $200,000 a year for monoclonal antibody therapy. And my argument was, we can deliver mRNA that encodes the monoclonal antibody. It uses the host cells as the factory to produce the protein. And there's a lot of amplification going from RNA to protein. So it'd be safer, it'd be easier, it'd be cheaper. And again, they would nod and they would say, get a real job. The next problem we had was, how do you deliver mRNA? So just about every cell and every fluid in every mammal's, every organism's cells contains RNases. That's an enzyme that degrades single-stranded RNA. If you inject uncomplex naked RNA into an animal, it's gone in seconds. It's degraded. So what we thought is we need some sort of carrier molecule. We studied about 50 different formulations and finally got our hands on lipid nanoparticles. Lipid nanoparticles that are used in the COVID-19 vaccine were actually developed to deliver siRNA, short interfering RNA. They're 21 base pair double-stranded RNA fragments that degrade RNA at specific sites. We had to figure out, could we use these same LNPs to deliver mRNA that's 2 to 14 plus thousand base pairs long and single-stranded? So we put luciferase mRNA into lipid nanoparticles. Luciferase is the enzyme in fireflies that makes them glow at night. So if you give a mouse luciferase and you give them substrate for the enzyme, the mouse will glow. And that's what we're seeing here. Systemic injections go to the liver. LNPs go to the liver. But peripheral injections, IM sub-Q, remain at the site of injection and are translated for upwards of 10 days. I want to now switch to what I think of as the future. So this is a statement from a conference on gene therapy. And I made the conclusion at the bottom to show you what 4 days of gene therapy seminars came up with. Their conclusion was location. If you can't get the delivery vehicle, the lipid nanoparticle, the virus, to the cells of interest, it does you no good. So my lab, again in around 2016, started to study how could we target lipid nanoparticles to things other than the liver. And we started off by studying endothelial cells, T cells, and bone marrow stem cells. And we took a very simple approach. The idea was that we could attach a targeting moiety. Could be a piece of an antibody, it could be a receptor ligand, could be a peptide, it could be just about anything that bound to cells of interest. And we attached them to the surface of the lipid nanoparticle. When we did this, the morphology, this is an electron micrograph of lipid nanoparticles before and after antibody. So the morphology stayed the same. They got a little bit bigger. That's the size of an antibody coating on an LNP. And most importantly is they maintain their morphology and their functional characteristics, surface charge being the most important. The first ones we made, we put an anti-PCAM antibody. PCAM is an antigen expressed by endothelial cells. And here we're simply asking, can the PCAM labeled LNPs bind to PCAM expressing cells? Yes. Could they be taken up and translate the protein? And it was only in the presence of PCAM targeting. That's shown here. These are primary endothelial cells from a human. And what we see is that in the absence of targeting, there's no uptake. There's no delivery of RNA to these cells. With PCAM targeting, 95% of the cells now express the protein of interest. Now, nobody cares about in vitro studies. So we then injected these LNPs into mice. And what we saw was interesting. The first thing was that when we added an antibody to the surface of the LNP, either control or anti-PCAM, we reduced liver uptake. That's because LNPs bind ApoE and use it to target the liver. And if you block ApoE binding, you block liver uptake. But the more interesting was that when we had PCAM targeting, the lungs now took up the LNPs and expressed the mRNA. We calculated that about half of the delivered activity went to the lungs. Now, I always like to mention CNS targeting because I'm always asked this. It's usually by the older folks like me in the audience, and you can think why later on. But I was a resident in Boston, and this was decades ago, many decades ago. Our biggest fear was the Friday night stroke admission. This would be often a person anywhere from their 50s to their 80s who suddenly developed weakness in a leg or suddenly had difficulty speaking. They were able to walk into the ER, and when you do a CAT scan on them, you see a region of brain death. This is a middle cerebral artery stroke. You admitted them to the hospital and you spent the weekend with them. The problem was what happened over the next few days. And you can see that here. What happens with stroke is you have dead brain cells that release inflammatory mediators. There's no room for things to spread in the brain. So what happens is you build up pressure and you kill surrounding brain. And you can see that here, nearly the entire hemisphere is now dead. These patients don't walk home and often don't leave the hospital. So we wanted to know, could we treat this? Could we develop an RNA-based therapeutic? Working with collaborators that had a system where they could micro-inject inflammatory mediators into one hemisphere of a mouse brain, we put in TNF, an inflammatory cytokine. We then treated the mice with LNPs that we had targeted to the blood-brain barrier, to the brain. And we delivered an RNA encoding thrombomodulin, which is an anti-inflammatory mediator. What we're doing here is we're measuring capillary leakage. And you can see on the other side of the brain, not much is happening. But on the side of the brain that got the TNF, untreated animals had huge amounts of capillary leakage. That's the inflammation from the expanding mediators. But when we delivered brain-targeted anti-inflammatory mediators, we got rid of all of the capillary leakage. When we went back and did a CAT scan on these mice, the brain infarct didn't increase. We're now translating this into similar therapies for humans. Now, I'm an immunologist, and my interest is always, of course, treating immune cells, treating the bone marrow. The question is, though, what advantages do you have from engineering lymphocytes in vivo? And there are too many to list. They start with things like making CAR T cells, which are very potent leukemia-killing agents. You can deliver agents that make killing better to not treat cancer or treat an infection. You can deliver agents that make killing worse to treat graft versus host disease or autoimmunity. So there's many different ways you can modulate immune function if you can target those immune cells in vitro. The difficulty is that with immune cells don't have endocytic activity. What that means is they can't take up particles like lipid nanoparticles. So our idea for targeting lymphocytes was, well, if we bind a surface marker on a lymphocyte and that marker endocytoses after binding, then that will take the lipid nanoparticle in. And that's what we see. So here we're treating 293 T cells on HeLa CD4s, and it doesn't matter if the LNPs are targeted. We added a CD4, anti-CD4 antibody and treated purified CD4 positive T cells. In the absence of targeting, nothing was taken up. But when we targeted the CD4, we had high levels of translation. We injected these LNPs into a mouse, and what we saw was that the spleen lit up with targeted LNPs. 20% of spleen cells are CD4 positive T cells. But we went back and we re-imaged the mice after we removed all of the organs on the previous page. And what we saw was that in the animal that got the anti-CD4 LNPs, lymph nodes, this is a para-aortic and inguinal lymph nodes, now lit up. So what that means is that the LNPs injected IV left the circulation, went into the tissues, into the lymphatics, back to the draining lymph nodes, where they could be taken up by CD4 positive T cells and the RNA expressed. And here we purified the T cells from the lymph nodes to show that's where the luciferase was. So this told us we could effectively deliver therapeutic RNAs to lymphocytes in vivo. We switched to an easy system, a Cre-Lox system. These are mice that have a gene, usually a fluorescent protein, with a stop codon in front of it, surrounded by LOX-P sites. If you deliver Cre recombinase, it recognizes the LOX-P sites and cuts out the stop codon. So it turns on fluorescence. And you can count fluorescent cells to know where the LNPs were delivered. We saw pretty good efficiency, 40 to 50% in both lymph nodes and spleen. This is actually the worst of all of the delivery technologies that we've used. So our initial conclusions from this work were that we could deliver LNPs encoding any RNA we wanted to specific cells and organs in vivo. That then led us to decide which of the thousands of different therapeutics did we want to work on. Now, I come from Penn, and as you probably know, Penn had the first two CAR T cell therapies approved by the FDA through work that they did. So we, of course, said, could we make CAR T cells in vivo? This is a picture of what a CAR T cell is. So this is a cytotoxic T cell. It has a T cell receptor that recognizes a viral or cancer-derived peptide in association with MHC, and it's a very effective killer. What a CAR T cell does is it replaces the exterior region with a piece of an antibody that recognizes a tumor-associated antigen. So the first ones that were developed bound CD19. CD19 is expressed by B cells from pre-pro through mature, and it's expressed by most B-cell leukemias and lymphomas. It's been put into patients for 15 years, and it has a cure rate of around 60 to 70%. These are in patients who would have died of their disease. So an incredibly successful therapy. The problem with it is what it takes to make a CAR T cell. You have to start by leukapheresing the patient. This is a big machine that takes blood out, spins it around, removes the white cells, puts the rest of the blood back, does that over and over and over until you get a couple billion cells out. You then have to take those cells that you just purified and infect them with a lentivirus. The lentivirus delivers the CAR gene. You then have to expand the cells for about 10 days to make enough cells to give back. You then have to give the patient high-dose chemotherapy, known as lymphodepletion chemotherapy, It essentially makes room for the CAR T cells to engraft and expand. Then you treat the patient. In the US, this costs about $500,000 a dose. It requires incredibly sophisticated hospitals that have the facilities to do all of this to make CAR T cells. There are very few CAR T cell capable hospitals in the low and middle income parts of the world. Sub-Saharan Africa has one. South America has two. Southeast Asia has one. China finally built one. Incredibly rare, and they're too expensive to use even if people had them. So our idea was, if we could target the lipid nanoparticles to T cells in vivo and encode the CAR in the mRNA, we could make CAR T cells in vivo. It would be a simple injection of frozen RNA LNPs, very easy to deliver, doesn't take 2 weeks to develop, certainly doesn't cost $500,000. So we started with a model of cardiac fibrosis. This is where you make mice hypertensive by putting pumps with different factors in them. And the hypertension causes cardiac fibrosis. This is an echocardiogram from these mice. So the gray are normal mice. This is normal heart function. This is the ejection fraction, how much blood is pumped out every time the heart beats. The brown is hypertensive, fibrosed heart mice. You can see the volumes of the heart at systole and diastole are increased. It's stiff, so it can't move, and the ejection fraction is greatly decreased. The red is a single treatment with T-cell targeted mRNA LNPs, and we returned heart function to normal with one treatment. This is a stain of the heart of cardiomyocytes. Red shows up as fibrosis. You can see the normal animal has very little. The hypertension regulate therapy and give 3 or 4 days of antifibrolytic or other treatments. We've now taken this to the next step, and we made a decision that we would use this therapy to target B cells. So in B cells I talked about earlier, we talk about leukemia and lymphomas, which are very effective therapies. But in autoimmune diseases, there are many autoimmune diseases that are based on B cell dysfunction and aberrant B cell responses. So the idea is that if you treat somebody with lupus or myositis or other inflammatory diseases with a B cell depleting therapy, you will erase their prior immune system and replace it with a brand new naive system that doesn't have all of those autoimmune B cells. And when you get rid of the B cells, that gets rid of the autoimmune T cells that require the B cells for activity. So it's a simple approach. We make mRNA LNPs with CD19 CARs, B cell depleting CARs in the mRNA. We deliver this right now, the studies I'll show you are to macaques, where the CAR T cells transiently deplete all of the B cells, and we see what happens. This is simply taking cells from a group of macaques and treating them with CD19. In this case, we use CD20 because it cross-reacts with macaques. targeted T cells, and we see that we can get around 80% of their T cells in vitro to take up LNPs and put a CAR on their surface. It's in a dose-response manner that's expected. We next injected these LNPs and a dose response into macaques, and we measured expression of the CAR on the T cells. And we don't understand this, but what we saw was with each dose, and we dosed every 3 days, we saw an increase in the number of CAR T cells made in vivo. So after 3 treatments, nearly 80% of the T cells in these macaques had a CD19 CAR on them. We treated macaques and followed them over time. The PBS treated macaques, nothing happened to their B cells, and their tissues remained full of B cells stained in black. At the lowest dose, we got rid of all of the B cells in the circulation. And as the dose increased, we got rid of all of the B cells in the tissues. So this means we nearly completely depleted all of the B cells with a single round of treatments with in vivo CAR T therapies. We followed these animals because we wanted to see what came back. You can see here when they started, they had a mix of naive and memory B cells. Around 3 to 4 weeks, and this is dependent on the dose, the B cells returned. And they initially returned as completely naive B cells. Over time, we started to see some class switch. We don't know, is this the naive B cells becoming mature, or whether there were some leftover B cells that we didn't deplete. What we're doing with this is now we want to move this into people, and we've already treated the first couple people to transiently deplete their B cell repertoires to treat severe autoimmune diseases like systemic lupus, myositis, and others. But the big difference between this and ex vivo lentiviral CAR is that lentiviral CARs have a mortality rate up to about 5% from the chemotherapy, from the adverse events of receiving the CAR Ts. So you can't use them on autoimmune diseases like type 1 diabetes or thyroiditis or rheumatoid arthritis, where the patients don't die of the disease. They just suffer from the symptoms. In vivo CAR T cell therapy should have no adverse events and no mortality. So after we treat our myositis and lupus patients, we're going to start treating type 1 diabetes kids and other more, I guess, moderate, not sure that's the right word, autoimmune diseases. We also wanted to see, could we target bone marrow stem cells? So there are thousands of genetic diseases of bone marrow stem cells. With sequencing, the number keeps increasing. But the bone marrow stem cells are responsible for all of the cells in the blood, the red cells, the white cells, the granulocytes, all of the macrophages in the body, except for the brain and lung probably, and a few other cell types. There are many genetic diseases as a result in bone marrow defects. The principal one is probably sickle cell anemia. 350,000 people a year are born with sickle cell anemia, principally in sub-Saharan Africa, but also in India. Mexico has a high rate. Numerous other parts of the world have high rates of sickle cell anemia. The current treatments are not great. They're characterized by painful episodes of clotting of the sickle cells. And we treat them with a variety of things. None of them are very good. We've extended lifespan to 40, but it's a miserable life. So our idea was, if we could target stem cells in vivo and deliver gene editing technology, we should be able to cure sickle cell anemia and be able to give people injections on the street to do it. So first, we wanted to target the bone marrow stem cells. Here we use CD117, which is a shared marker between stem cells, progenitor cells, and a few other lymphocyte populations. But you can see we had about 80% efficiency in targeting stem cells. We treated the Crelox mice I talked about before and followed fluorescent cells over time. And you can see that after about 4 weeks, most of the red cells were gene edited. Lymphoid, myeloid, other cells at different rates depending on how quickly they're replaced. But we had about 70 to 100% editing efficiency. Bone marrow people don't believe this data. They want you to do something called a secondary transplant. That's where you take another group of mice, you irradiate them, or you give them high-dose chemotherapy to kill their bone marrow, and then you transplant bone marrow from your treated mouse. So we did that, and we waited 6 months after a single treatment and transplanted the bone marrow cells into a new mouse. And again, all of the red cells, white cells, and granulocytes came from the gene-edited stem cells. So we had edited repopulating bone marrow stem cells at an efficiency of about 75+%. Sickle cell, you probably need to treat 25% of the stem cells and correct the gene disorder. So if we can target 70%, that should be doable. There are many other things that you can use bone marrow stem cell therapies for. We're looking at using it as a way of killing stem cells for bone marrow transplants. Currently, we use high-dose chemotherapy that has a mortality rate and a high morbidity rate. If we can use LNPs and selectively kill them, that will reduce the toxicity. You can deliver gene editors for thousands of genetic deficiencies, SCID, thalassemia, the list goes on and on. You can deliver therapeutic proteins to stimulate granulocytes, platelets, red cells, whatever you need. We've started with the base editing to fix the sickle cell defect. This has been published by other people, but here we're delivering it as an mRNA LNP targeted to bone marrow stem cells. You can see here, this is red cells from bone marrow of a person with sickle cell. And all of these arrows, you can see sickle cells. This is a low dose and high dose treated. You can see here, we've gotten rid of nearly all of the sickling with a single treatment. So one treatment in vitro of bone marrow stem cells with our gene editor delivered by RNA LNPs looks like it may work to cure sickle cell. We're now testing this in macaque models and hope to move to people soon. So in my view, RNA has an enormous number of potential therapeutic avenues for development. We have our ID vaccines, flu, COVID. We have vaccines for cancer. We've already shown efficacy in pancreatic cancer that nothing is able to treat. We have vaccines for food allergies and environmental allergies. We're making vaccines for autoimmune diseases. We can deliver therapeutic proteins that treat stroke or heart attacks or other heart defects. We're now working on delivering to central brain, not just the blood-brain barrier. So it might be useful for Parkinson's, Alzheimer's, ALS, and other congenital other brain diseases, as well as congenital deficiencies that mark a lot of the epilepsies. So I wanted to quickly end by talking about my program in vaccine and therapeutic equity. This has been a lifelong career decision, approach, interest. It came to a head during the COVID-19 pandemic when the RNA vaccines were released. The US, Europe, and a few other countries that could afford it all had RNA vaccines in a matter of weeks and had completely vaccinated their populations in under 2 months. The low and middle income countries, sub-Saharan Africa, Southeast Asia, it took over a year and nearly 2 years for them to get any access to RNA vaccines. This is a distribution of the population in the world. The majority of the population is in sub-Saharan Africa and Asia. This is a distribution of the money. The majority of money is North America, Western Europe, and a few other scattered countries. This is a distribution of malaria. It could be TB, it could be sickle cell, it could be a variety of other diseases. As you can see, the diseases are where the people are, not where the money is. That's problematic because pharmaceutical companies have no interest in spending hundreds of millions of dollars developing therapies for regions of the world that can't afford the therapy or the vaccine. So what that means is these regions are deficient both in access to advanced medicines and therapeutics, vaccines that they need. I started working with Thailand, a university in Thailand, about 10 years ago. This is before anybody knew anything about RNA. But Kiat, shown in this picture, who runs the Chula VRC, heard my lecture and brought me to Thailand to talk to them. And during that visit, we arranged to bring mRNA technology and make it the core of the Chula VRC. When the pandemic hit, I got a call from Kiat, and we spoke about once a month, who's around, it was probably March of 2020. And he said, I've been talking to my government, to my health minister, to the royalty. We're concerned that any vaccine made in the West will take years before it ever makes it to Southeast Asia and other low and middle income countries. And they weren't willing to wait. So Kiat talked to the royalty, the government, donors, and got money from them to make their own vaccine. So Kiat and I worked together and we made a Thai version of the RNA LNP vaccine. This is a phase 1 clinical trial showing these results. What you can see, this line, this is the results from the Pfizer-BioNTech COVID vaccine. So the vaccine that Kiat and I made gave better, higher titers, better results than the Pfizer vaccine. I don't show this slide when I talk to Pfizer. But we had another problem. We didn't have a way of making GMP product. So GMP is the type of manufacture that you need to make vaccines and therapeutics and drugs for people. It's a highly specialized negative pressure environment, gloves, gowns, masks, tanks. It's multi-million dollars to build and multi-million dollars to run. So we got money from the royalty and we built a GMP site in Thailand that's shown here. Around the same time, I started working with the WHO, and I convinced the WHO that this was a good way to spread equity and technology throughout the world. And they agreed to build 15 GMP sites in low and middle income countries around the world. There's a few in Latin America, Sub-Saharan Africa, Southeast Asia, Eastern Europe. We even built one in Ukraine in the middle of the war. How, I don't know. So the idea behind this is that by giving every country in the world access to what I think is the newest upcoming technology, you give them access to developing therapeutics that they need. Big Pharma isn't going to make another dengue vaccine, but Kiat has a dengue vaccine in clinical trials right now. He has 9 other vaccines for dust mites, for tularemia, for HPV, and a bunch of other diseases that are mainly in Southeast Asia or tropical regions. In Sub-Saharan Africa, they're working on malaria and TB vaccines. And we're working with Brazil right now on a Zika vaccine. We've also arranged with the Penn patent people to give access to all of the technology that we patent to low and middle income countries for no licensing fee. So they have access to all of these vaccines and therapeutics that will help them kickstart their programs as we develop basic science research so they can make the next vaccine and the next therapeutic. So I have to thank the people in my lab who made these studies possible and all of my collaborators, and I'll stop there.
Mil gracias al doctor Drew Weissman por esta espléndida conferencia. Y a continuación, las doctoras Ana Carolina Sepúlveda y la doctora María Soledad Funes harán entrega de un reconocimiento por esta espléndida participación como conferencista.
Thank you very much. Thank you.
Asimismo, le hacen entrega de presentes por su visita a nuestra facultad, a la Universidad Nacional Autónoma de México. Y ahora les vamos a pedir muy respetuosamente a nuestro invitado especial y a las doctoras que lo acompañan a que pasen a la primera fila de este auditorio para obtener una foto grupal con todos los presentes esta tarde. Por favor. Les vamos a pedir a todos ustedes si nos acompañan poniéndose de pie. Y de alguna manera acercarse al centro, cuidadosamente hacia el centro, para ocupar los pasillos hacia el centro del auditorio, y nos preparamos para tomar esta fotografía que seguramente será muy importante y estará publicada en las redes sociales de nuestra facultad, así como en la Gaceta de la Facultad de Medicina.